Welcome, welcome. Can you hear me and see the welcome slide? Hopefully, okay. Welcome. Yeah, this is a very exciting and key meeting for us, our first advisory board meeting. I truly believe that these interactions and brainstorming will substantially enhance the impact of this project that is essentially aimed at enhancing the sustainability of our rangelands. I thank all the co-PIs for your efforts, talents, and expertise and interactions that led to this successful project to initiate. I also thank all the advisory board members for your willingness to help to be part of this group since day one without hesitation. I am very happy to see, well, now so many names, not some pictures, but so many names and faces with whom I have shared many significant and fun parts of my professional career. And I'm so happy that this project will give us a chance to work together one more time. For those that I still don't know from the advisory board, looking forward to start interacting with you. Thank you again for your willingness to share your insights, talents, expertise during this meeting and also into the future. We are very lucky to have you on board on this advisory board. So we try to make this a really dynamic and hopefully interactive meeting, and for the years to come, try to make it live. But let's start with introductions. We'd like to introduce our advisory board members. I did that in alphabetical order. Just to give me your name, organization, and expertise, like a really quick introduction. And then we'll do the introductions for the co-PIs as we are presenting, so we make it more dynamic. So the first one that comes in I see that you are there, Tim. Great to see you, and you see your name there. So let's start with introductions. Yeah, and then Tim Bateman, really glad and happy to have you on board.
Hi, me too, Juan. It's good to see you, and
good to see you.
I've worked with, so I'm excited to be here and see where I can help out. And I'm honored, that you asked me to be a part of this. So, my background is I was a master's student of Juan's from 2015 to 2017. We looked at the spatial and temporal dynamics of Medusa Head in Eastern Washington using remote sensing techniques. And Doug was a committee member on that project as well. Very successful project, and we were very happy with the findings there. So after that, I've spent kind of 5 years working as a remote sensing analyst for Open Range Consulting. We developed remote sensing based products for Western landowners, and was very happy to be a part of that as well. So currently, I'm actually a employee of Colorado State University, but I'm based in Louisiana. So the organization on that in the introduction is kind of misprinted, but that's okay. I work for the Center of Environmental Management of Military Lands as a GIS analyst for them. And I offer GIS support for Air Force bases in Louisiana, Mississippi, and Alabama. And then also I currently operate a GIS kind of consulting company called Western Skies GIS, where I offer similar support for Western ranches, landowners, and land managers to collect, develop, and manage some of their infrastructure layers and environmental GIS layers, and then help develop communication tools for them. So again, I'm happy to be here, happy to see where I can help out. So with that, I'll pass it on to, looks like Rick Denver.
Yeah, Rick.
Sorry if I missed.
Yeah, hi, Rick, are you there? Okay, so yeah, so we'll have next tomorrow also for introductions for people that are not here now. Let's move to Corey Forbes.
Oh, Rick's here. You're on mute, Rick.
Oh, Rick is here.
Can you hear me now?
Hey, Rick, how are you? Great to see you.
Good, good. Sorry about that. Yeah, my name is Rick Danver. I've worked quite a bit with Juan and Fred and Beth and the group over the years. I used to be the wildlife manager for Deseret Ranches. So we worked a bit on projects up on a Deseret Land and Livestock Ranch in northern Utah. I've always been really enjoyed the work we've done with this great group of people. Now I'm largely retired. I do a little bit of work part-time with private landowners still, with wildlife and range issues, and also work part-time with the Western Landowners Alliance. So I'm just happy to be here, and I'll pass it on to Corey.
Great to see you, Rick.
Good to see you.
All right, thanks, Rick and Juan. I appreciate it. Hi, everybody. Good morning. My name is Corey Forbes. As the slide indicates here, up until this summer, I was an associate professor of science education at the University of Nebraska-Lincoln, also serving as coordinator for Institutional Science Literacy Initiative and director for the National Collaborative for Research on Food, Energy, Water, Education. Actually, since August 1, I have moved southward. I'm now a chair of the Department of Curriculum Instruction at the University of Texas at Arlington and professor of science education here. My work has generally spanned sort of the science education and ag and natural resources education worlds across, and I do work in K-12 and undergraduate and graduate education settings. So I'm also super excited to be here. I know a couple familiar faces with Kathy and Wei Wei, and I believe Sarah's on here as well. But otherwise, I don't know the rest of you, and I look forward to working with you.
Great, thank you. Yeah, Sarah. I think I saw you.
Yep, I'm here.
Okay.
Hi, everyone. I'm Sarah Haynes. I'm a science education and biology professor at Towson University, which is just north of Baltimore. I teach courses for the elementary ed students and secondary ed, and also some environmental education courses for our environmental science majors. And Chris, it looks like you're up next.
Hi, Chris.
Hi.
Yeah, great to see you.
Yeah, good morning, everyone. I'm Chris Holby. I am the lead scientist at Working Lands Conservation, a small nonprofit in Logan, Utah. Do a lot of work on ecosystem services across rangelands, and have a background working on Forb Islands as well. So really happy to be part of this collaborative in this effort. Thanks, guys.
Great, thank you. Great to have you. Emily, I haven't seen your name. Are you around?
Yep, I'm here, Juan.
Hey, Emily, great to see you.
Yeah, great to see you. It's been a while.
It's been a while.
Yeah. Well, so I currently work for Colorado State University, but I was a student of Fred's back in, I guess, 2008 is when I graduated. And so Juan was so kind as to also be on my graduate committee. And yeah, so my background's in livestock behavior and animal science. And then I've worked for extension in Colorado and Montana. And I work at a research center right now in Colorado, but I'm actually transitioning to go back to extension starting in January. So I'm excited to be on this project, and it's great to see some familiar faces.
Great. Happy to have you on board. Then Ashley.
I'm Ashley Longmore. I was a student, a master's student of Fred and Juan's. I got my degree in range sciences and have worked for the state of Utah for the Department of Agriculture with the Grazing Improvement Program going on 10 years this coming year. So I work with ranchers in the Wasatch Front area. So I cover Tooele and Utah counties and Wasatch, Summit, Morgan counties, and we work on helping ranchers improve their grazing programs by being a cost share grant program for implementing infrastructure, mostly lots of water systems, solar systems, fence lines, seedings, things like that. So I'm happy to be here and excited to see a lot of faces.
Great. Yeah, great to have you, Ashley. Great to see you. Yeah, now Brody Mong is our next advisory board member. I saw your name, but Brody's somewhere.
I'm here. Anyway, good to see you again, Juan. It's
Likewise.
been a while, that's for sure. I'm a former, master's student of Juan's, technician of Fred and Juan, and also Jennifer McAdam here. I see worked with a lot of people here on this list. Yeah, it's been a little while ago. Currently, I am the farm manager at Bruce Seed Farm. We're located in Townsend, Montana. It's not your typical farm here. We're located western Montana along the Missouri River here in the valley. We raise forages, reclamation grasses, all kinds of different species used in projects like that. We're currently raising about 27 different crops and varieties here on our farm, and I oversee the production of all that. Let's see, many of the species that are involved in our project here as well. We sell and market many of all kinds. We have access to all kinds of species used in projects like this, other reclamation projects, grazing projects as well. So anyways, I'm just glad to be invited to be a part of this, and looking forward to it.
Great to have you, Brody. Yeah, and we have some seed from you already, so.
Yeah, that's right.
Yeah, so Spencer Morris. I think I saw
For some reason
name.
can't get my video to start here, but I'll just go anyway. Spencer Morris. I live out in Park Valley. I'm a 5th generation rancher here, and I've served on the West Fox Conservation District, where I got to rub shoulders with you guys like Eric Thacker and Travis Moe, Jay Tanner, and learn from their expertise. And I'm grateful for the opportunity to be able to be here with you guys, and hopefully I can contribute more than I learned here. So thank you.
Great, Spencer. Great to have you on board. Travis Moe. Travis, yeah.
Good morning. I don't know if my video's working or not. I can't see it, but I'm Travis Mote. I worked with Juan almost 20 years ago coming on.
Time flies, huh?
Yeah, that's been a while. We worked on diet sequencing with tannins and terpenes. Since then, I've been a range con with the NRCS for about 12 years, and now I'm currently the assistant state conservationist for programs for the state of Utah for NRCS. And I'm glad to be here and excited. Thanks.
Great to have you, Travis. Yeah, great to see you again, or listen to you again. Bray Owens. Are you, haven't seen your, are you muted or? Okay, so we move to, yeah, as I say, we have a chance to introduce you tomorrow. So Shake, I know Shake is Shake Owens. I saw your name and you sent an email this morning.
Juan, this is Kathy. While we're waiting, we skipped Beth Klein up in the K. So if Beth could introduce herself now.
Oh, okay.
That'd be great. Thank you.
Oh, yeah, I didn't, yeah, sorry about that. I didn't see the sequence. Yeah, Beth, yeah.
Thanks, Kathy. Thanks, Juan. Yeah, so Beth Klein, I'm a professor of science education at SUNY Cortland. I'm also the campus sustainability coordinator. And in those roles, I do science education for our early childhood and childhood ed program. And we incorporate outdoor education as a large component of that, including garden space. And our current project on campus is the development of an edible food forest. Also, a founding board member of the Truxton Academy Charter School, which is the first ag-focused elementary school in New York State. So I work in that as well. So happy to be here. Glad to see a few folks I know, Kathy and Sarah and Rita. I'm looking forward to working with you all.
Welcome. Yeah, welcome.
Well,
And then
we'll try it again. Can you hear me now?
Yes. Jake?
Okay. This is Jake Owens.
How are you?
Sorry about that one. It was trying to get me on a different microphone, but apparently I'm on the right one now. So
yeah.
Nice
to see you.
Yeah, glad to visit with you. I worked with Juan and Fred years ago on some biochemical interactions with some grazing forage. It was a good project. I enjoyed it. Since then, I've worked for the Natural Resources Conservation Service as a range con for a handful of years, and now I'm in eastern Idaho, along the border of Wyoming and Montana, and several places in between. A diverse landscape. So it's going to be great to hear what you guys are doing and kind of about this project. I think it'll be applicable to the area that I'm working at, and glad to be here.
Great to have you. Yeah, Shake. Yeah, and then Fred Provenza, I saw your name, I think.
Yeah, I'm here, Juan. Wonderful to be with all of you and to see all these faces from out of the past. I'm retired, been retired for 12 years. Prior to that, though, spent a wonderful 35 years at Utah State University working on behavioral ecology of plants and animals and working with all these wonderful students and with Juan for many, many years. So looking forward to participating with all of you.
Great to have you, Fred. Yeah, that's great to see you. Yeah, the next, Taylor Payne. Yeah, Taylor, I think I saw your name also.
Yeah, how are you? I'm glad to be here with everyone too. I'm also a student of everybody there from Utah State. So hello to you all. I am living in Randolph, Utah, and I coordinate a region of the state of Utah doing grazing improvement projects. And I also have a consulting business doing range monitoring. So I'll be excited to work with you today. So,
great to have you. Yeah, great to see you. Shay Tanner.
Hi, this is Jay Tanner. I live in Grouse Creek, Utah. We ranch in northwestern Utah and northeastern Nevada. I'm involved in several conservation programs in the state of Utah and also involved in a national group. I'm a director on a group called Foodscapes, who work for improving collaboration with private landowners and public land managers. Look forward to working with you.
Great, great to have you. Yeah, welcome. Kathy both.
Hello, can you hear me?
Yeah, great to see you. Yeah.
Yeah, good to see you too. Well, you can tell I know Juan. Basically, I used a lot of the research that Fred and Juan and all the folks there at Utah State University were doing to figure out a method for teaching cows to eat weeds. And since I spent a lot of time figuring out how to do that, I realized that it's hard for regular folks to understand research and figure out how to use it on their grazing operations. So I currently am the publisher and editor for an online grazing magazine called On Pasture. And I translate research and experience into practices that graziers can use to be more sustainable and profitable. So I'm looking forward to participating. Thanks for having me. And it's nice to see everybody. Thanks.
Great to have you, Kathy. Yeah, great to see you. And then Hui Wang, our last advisory board member. Are you?
Yes, I'm here. Yes.
Great.
Good morning, Juan. Good morning, everybody. My name is Hui Wang, and I'm based on the Purdue University. I have a joint appointment with curriculum instruction and agricultural science education communication. Mostly right now, my works relate to integrated STEM teaching methods through agricultural for natural resources. So that's kind of like what I'm doing. And mostly I don't know everybody here. And the only person probably I know that Corey already shout out. Hi, Corey. So very nice to meeting everybody and happy to working with everyone. Thank you.
Yeah, nice to meeting you. Welcome. Okay, so yeah, that's as you see, there's a really diverse group of people, backgrounds, locations. I think I'm, yeah, really it's related to this project, not related to diversity and biodiversity. So I think it's great. So yeah, I will just start with a really brief overview of how this project started, or the idea behind it, I guess. So, and that's where we have the problem here, right? So normally seeing cows grazing on a sea of monotonous grass, that's the issue that we normally see on rangelands, and even more as we move into the future, we'll see this pattern even more. And even worse, this is a picture for a site we're working on Washington State in the Eastern Washington in the Scablands, and then see those cows grazing on a sea of Medusa head. So that's the situation that these animals are under, right? So challenged by this monotony, and also by these by grasses, and the food value or the feeding value of those grasses also drops, in the midsummer, disrupting many different nutrient cycles, from providing supplements, animals being inefficient, so enhancing the emissions of methane, reductions in nutrition, reductions in animal welfare, and in general, reductions in biodiversity. But if you look at, from the other side of the coin, these animals evolved in the Mediterranean region, ruminants, grazing a diverse array of broadleaves and grass species. And, that's how this Larson depicted those cows. Of course, 65 million years ago, we didn't have flying cows, but we have cows. What he really hit the point is that eating not just grass, but a diversity of species, those broadleaf species that had several benefits, as we'll see, to the animals, right? Not just eating grass. So how then we break this monotony? And that's the idea that came out. Well, why don't we create these landscapes where we see islands in this sea of grass? So try to provide those islands in this sea of grass, creating this with legumes, forbs. And then one of the benefits of those legumes is that they provide a broad array of diversity of chemicals, mostly, for instance, carbohydrates and protein, that enhance, for one, the nutrition of the animal, and also provide synergisms that enhance the nutrition of the animal and reduce environmental impacts. On the other side, those broadleaves and forbs and legumes contain these bioactives of these secondary compounds that we know that many of them evolved to defend the plants, so being like toxic, but then at appropriate doses, these plants, like some of these are the ones that we are using in our project, right? So these broadleaves or legumes or forbs, they provide antiparasitic, bactericidal, blood attenuation, antioxidant, medicinal effects to the animal, and also reduce environmental impacts like the emissions of methane and ammonia from the animals. They enhance soil health, and we are also exploring that avenue, looking at how that enhances the microbiome on the soil from these secondary compounds. And also they have compounds that improve meat and meat quality. So this is a master plan that we have, is that those animals being in those islands, we can enhance productivity, enhance health and welfare, reduce, modify animal distribution, and also reduce and enhance fecal nitrogen, reducing urinary nitrogen, creating these functional landscapes that not only provide chemical diversity, but also structural diversity, connectivity, habitat for wild species, and also for pollinators. So the benefits are just, or the services are beyond just the nutrition of the cow, but providing, structure and connectivity across landscapes. So that's the master plan. And then we have research, extension, and education components that, the USDA allows us to, I think, in a great idea, trying to make this an integrated project. So I think the challenge we have, I think we're starting with this meeting, is trying to integrate these components, research, extension, and education, in a way that this project becomes a living entity, right? With not just compartments completely separate from each other, but just a living and breathing entity that is articulated among these different components, right? So that's the challenge, creating this, essentially this living organism among, PIs, students, which I think could entail, the blood that is flowing through these connections, other personnel, and then the advisory board, you guys that providing us, with feedback in terms of when we presenting these ideas, hopefully showing or suggesting strengths, challenges, opportunities, as we'll come back to these three topics, but that's, the feedback that ideally would like to discuss and brainstorm with you. And at the same time, we're providing ideas and potentially into the future research and results that, you can use for developing new applications in your positions, the way you're working, new ideas. We can also, imply this, the advisory board also helping us disseminate these ideas and engage potentially, new members, new people that want to join and enhance the capacity of this project. So that's the overall idea, that hopefully if we can achieve this flow of ideas and integration across these components, I think that'll be something that at least we start developing here during this meeting. So the structure, I think, of the meeting is going to be fairly straightforward. Like we present to the advisory board this project in 10-minute presentations through this live broadcast. Then we have 5 minutes only for questions and answers, so we make it more dynamic. You can type your Q&A feature, type your questions or your comments. I may be able to read a couple of those, but then we'll keep, everything. We'll keep those comments and questions and ideas to discuss in these open floor discussions that will happen after each component of the meeting, right? So after the research, extension, and education. So we'll have this open floor brainstorm sessions, and then on day 2, we break out into rooms for research, extension, and education. And then again, we move forward to the open floor again to brainstorm and have new ideas. And so ideally making this a more dynamic, and hopefully into the future, we can follow this structure live as opposed to just through Zoom. So that's, so the way I want to start now is like, we have this master plan for the project, as I was showing you before. And within this master plan, the first thing that we need to accomplish is essentially, I've been talking to some of you about this, like some identifying species for these islands, which ones are going to be the ideal ones to provide this chemical and structural diversity. So that'll be the first issue to tackle, and then we'll move through this master plan. Hopefully we can fill this puzzle as we move forward through this project. So yeah, our first speaker then for this objective is Jennifer McCallum from Utah State University. So she'll be talking about this component, that I think is key to plan or devise the species that we want to seed in these islands, and maybe even in different ecoregions. So that's, so let's go ahead, and I don't know if Jennifer, you can start. I'll just stop sharing.
Thanks, Juan. I'll start sharing my screen here. Okay. Morning, everyone. It's nice to see some familiar faces and nice to see new people on the advisory board that I have yet to meet. So I'm looking forward to that. I'm a plant physiologist here at Utah State University, and my role on the proposal is to investigate plant species that are going to be used in the resource islands doing small plot work. I thought I'd introduce myself by talking about a study that I'm engaged in right now. We're looking at the yield and quality and tannin concentration of 6 different sandfoin cultivars. They're listed on the right side of that photograph. And the photograph shows the plots this spring. So you can see that the sandfoin is going strong. This was planted in 2018, and we've been sampling it in the succeeding 3 years. And we sampled it for yield at full bloom. The photograph here on the right shows a cross section of a sandfoin leaf. Juan was talking about some of the secondary plant products and those kind of corn grain-like shapes that you can see here in black. Those are tannin sacs. So the plant accumulates tannins in the foliage especially. And so that's one of the chemicals I've been working with, bioactive chemicals in plants. And that's one that we think we already know quite a bit about what it can do. So we'll be interested to explore it in other species. I just thought I'd show you the yield of sandfoin since it's a little bit similar to the work that we're going to be doing. This is dry matter yield in megagrams per hectare. And so if you want to convert that to tons per acre, cut the numbers about in half. So I've got these in order of increasing yield over the 3 years. Remont is actually the cultivar we're using in our study. And you can see what happens to the yield. Went from about 4 tons per acre in the first year down to about half that in the 3rd year. So one of the things that we want to do is look at the persistence and the yield over time of these prospective plant species. We looked through numerous sources when we were collecting the species that we were going to use in this study. One of the things that we kind of screened for was their protein and their feeding value in the second half of the summer. So these are just a couple of examples of sources. The NRCS has a plant guide, I think, for every one of the plant species that we're using currently in the study. So you can see the variety here, lots of good information. And this is the list that we're using. So we'll probably look at this again before we're done with this meeting. The grass that's included as one of the 24 species is crested wheatgrass. Everything else is either a forb, a legume, or a non-legume forb. And you can see the botanical nomenclature and then the reported bioactive chemicals, so quite a diversity. And as I said, I've done a lot of work with condensed tannins as well as with hydrolyzable tannins, and they both have great value for livestock production, we think. I thought you might be interested in the variation in seed size. I'm sure Brody's aware of this, but it's been an interesting challenge to get some of these calibrated for the planter and planted. Utah sweet vetch, this little container is about 2 inches across, and Utah sweet vetch is one of our larger seeded plant materials. You can see alfalfa here by contrast. This was interesting blanket flower. It's a little dense conical seed with a little tuft of hairs at one end. And this is one of our smaller ones, Western yarrow. So interesting challenges to work with these plant materials all in one spot. Our initial small plot study is at the Godfrey Dryland Farm near Clarkston, Utah. So it's convenient. It's just about 20 miles up the road. And there has been quite a lot of work done to understand, especially the soil at this site. So the image on the right is shows the outline of the Godfrey farm, and those dots, numbered dots, are the locations where soil was taken down to 3 feet and looked at extensively for the nutrient concentrations. There's no salinity problem there. Organic matter ranges from a very low 0.3% up to a very high for the West, 4%. Typically 2% is what we see in agronomic soils in Utah. And the pH is where we would want it to be for our legumes. But we did use the information, and we consulted with the extension soil specialists that wrote that publication to bring the soil that we're using there at the Godfrey farm into an adequate range for phosphorus, nitrogen, sulfur, zinc, and iron. We also added mycorrhizae, although we know that there will be mycorrhizae already present, but this is one that is formulated for herbaceous forbs. And so we thought that we wanted to provide everything we could to make this a success. But you can see the formulations of the fertilizers that we used. And of course, we also inoculated all of the legumes. These are just a couple of the inoculants that we used. And this is another satellite view of the Godfrey farm. You can see the outline here. It's about 30 acres, I think. And so our plots are located next to the highway that runs northwest along the side of the farm. There is a snowbank that was being installed the day that we flagged the plots for planting. And we have 5 replications, and each one with all 24 plant species. And so this represents roughly where the 5 replications are, and the plots are planted across. I think you can maybe see that better on the next slide. My tall assistant and I were out there. And so you can see the flags on this side. Here's the snow fence and the area where the snow will accumulate over the winter. And there's another row of flags coming down the other side. So if I show you this is the day that we planted, which was November 18th, using a Great Plains no-till drill. So each one of our, so you can see the 2 yellow flags here and 2 yellow flags over there. So the plots are about 8 feet wide and 100 feet long. And let's see, I think, yes. So Juan suggested that I make my little list of things that might be good discussion points. We're certainly open to alternative legume and non-legume forb species to include in the studies. I'm curious about your input on the use of these resource islands by grazing wildlife in addition to the cattle. I'm going to learn a lot, I'm sure, about how ranchers are going to manage cattle to best utilize these islands, and any additional data that you think we could be collecting while we have these small plots. They're a huge investment of time and effort, and so we want to make the most of them. We're going to be collecting data on plant density, yield, persistence, and then of course the type and concentration of secondary plant products. And then these plots are also going to provide material for the in vitro study that you'll hear about later. I'm going to stop sharing my screen. Thank you.
Yes. So yeah, I think we're still a little bit okay on time. So if you have any questions or comments, or I don't see anything still on the chat. Let's see. Okay, so yeah, the question, yeah, Chris is asking, what is the balance of native to non-native species in the assemblages? Were species chosen to have particular traits? So how you pick up those species?
I'm not sure if we're at anything close to 50%. We didn't have a particular target for the number of native species that we included. We know that some of the most reliable species are going to be non-native, like alfalfa, small brunette, sandfoin, and so that's a big part of what we want to learn. We want to learn what the secondary compounds in the, especially I'm of course interested in perennial legumes. That's what I work with mostly, but small brunette has turned out to be a really interesting plant. It's not only got hydrolyzable tannins that move nitrogen away from urine and into feces, which is beneficial to the environment, but it is jam-packed with starch. And so it's the candy, and we've used it in irrigated pastures, and animals will rush over to the little patches of small brunette that we included in each pasture and ignore the bird's foot trefoil, which is really rich and a wonderful plant, and they'll eat all of the small brunette. So there's so much to learn about these individual species.
Yeah, and then Jennifer maybe stressing that there's going to be other ecological sites where these plots are going to be seeded, right? That's in addition.
Yeah, we included 3 sites, and so tentatively in the proposal we suggested different elevations, but we certainly want to locate these in relevant areas that are typical of Utah grazing lands, and where we know we will have different environments from the one here at Clarkston.
Yeah, and potentially using different species, so that's still open, right? For discussion.
Very much open for input. Yep.
Yeah, so and then another question, and then Caitlin, I think, is asking related to things we measure, keep track of the bloom times, and then for different forms for pollinator impacts. So that's for sure something that, any comment there, Jennifer?
Well, keeping track of bloom times is something that wasn't at the top of my list, and I think it's tremendously important. And we would certainly be open to collaborating with graduate students of other folks who can keep track of the pollinators and that sort of thing. I think it would really add a lot of information that we could use.
Perfect. Yeah. So yeah, and then we'll keep these other questions for the, I think Angie is going to keep those for the open floor discussion later on. So, and now we'll move to our second component, that trying to start filling this puzzle. So in terms of looking at these species, now we want to see how they interact, or in terms of their synergisms with rangelands, right? So with the grasses that are already present in the range. So one of the approaches is to test these synergisms and complementarities in silico. So that's where it comes, my good friend Pablo Gregorini, that he's from the Center of Excellence on landscapes in New Zealand. So Pablo, is great to have you, and then he's going to talk about how the chemistry of these species is going to interact with the grasses in rangelands and we've been visiting about this already in this modeling exercise. So Pablo, go ahead. Yeah, so that's...
Well, good morning, everybody there. very good morning here. It's 5:45 a.m. of tomorrow for you. Juan has kept me awake for a while. So that's good. I already drank several cups of coffee. But yeah, like I was telling Juan, you know, farmers around me, you know, I have been awake for 3 hours already. So it's good to see some people that I know. Juan, Jennifer, I visited with you a while ago when I visited Utah, and also see Fred online. So it is great to participate in this project as well. And here in New Zealand, we in our Center of Excellence, you know, we are doing similar things at the landscape level. So it's a pleasure. So one of the things, let me share the screen. Can you see the screen?
Yeah.
Good. So as Juan was mentioning before, we've been talking about how we can use some techniques to evaluate these smart islands and then the plants that are part of these smart foodscapes in those islands, and you know, from a nutritional point of view. And so we thought of using a good old model, it's called MINDI, to simulate or to evaluate some of those processes and patterns of the grazing ruminant, you know, exploring or going to that island. So what I'm trying to do here in a very, very quick, although I've got a lot of slides and the presentation will be available for all of you, is to describe a little bit the model so we understand who are we dealing with and why, you know, the model that we picked in this situation, you know, is able to simulate some of those processes and the patterns of those processes. And then I'm quite keen to respond or answers regarding to the functioning of the model from a biochemical point of view and how we are trying to cope with some of those challenges. You know, so Mindy is, who is she? Well, it says it's a dynamic and mechanistic model that represent a dairy cow or a grazing ruminant. So the idea of using MINDI is because in vitro is quite complex and expensive to deal with some of the processes and patterns that we are trying to evaluate here. Using this particular model, and again, it's a dynamic and mechanistic model, deterministic though, enable us to look at intake, grazing behavior, rumen function, metabolism at the same time. And those are technical limitations that are posed into real life situations. So using this particular model will help us to advance quite a lot and the understanding of the dynamics of herbivores and those forages in those islands. A model per se is a representation of reality. You know, like the rule of thumb, all models are wrong, but we are trying to get close to the reality as much as we can. You know, since this representation of reality and this simplistic view of that complexity allow us to test hypotheses very quick and in a best situation. So the idea of using this model as compared to other predictive models is get closer to the reality possible, you know, from simple equations to going through a rumen models and end up in this model MINDI, which is a grazing ruminant. So this model MINDI, which is named after my wife, and represented in pretty much 8 years of my life in my previous job, it's just composed by 7 models. It's a cluster of models. with a modification version of the model Molly of Lee Baldwin. And then that interact with the sword canopy and structure and herbage quality or chemical composition model. It has a model of ingestive behavior, a model of feeding motivation, a model of preference and selection, a model of foraging bioenergetics, and last but not least, drinking and urination model. By the time I put this presentation a while ago, we were working on an oral processing model. So the model contains, pretty much an 8th model and a module in MINDI, which relates to a dynamic representation of oral processing and digestive output rate, which, as you can imagine, has a lot of influence in the way we simulate and manipulate in the model diets to see the impact of nutrition in methane production, for example, and nitrogen excretion. I'm not going to go through the details, but in a very simplistic way, I like to show the structure of the model. This is the core of the model, which is based on 3 main modules within the model, which is from an incentive point of view of that animal. So it's external stimulation systems. And then on the right, you'll see 2 smaller boxes that are internal stimulation systems, long and short term. And all these 3 systems, at the very end of the day, stimulate that motivation to eat and to graze, and the interactions and that motivation, determine the actions and the strategies of Mindy, the cow, to go for those particular feeds that are available to them. So going to, really fast in some of the internal modules of Mindy, we can represent the source structure, and we can represent the difference in biochemistry in the source structure in 4 dimensions, and mean, the 3 typical dimensions, plus the effect of diurnal fluctuations in chemical entities from a plant point of view, just at the old part of the plants, if you like. So based on that structure of the canopy, and based on the chemistry of that canopy and the part of that canopy, we mainly simulate by features as well. And throughout the day, Mindy simulates the diurnal pattern of grazing, which is, an interaction of those 3 modules of stimuli motivating that particular behavior, which is grazing. So we can simulate diurnal grazing patterns in addition to rumination patterns and eating patterns. So we Mindy can simulate those behaviors from actions and of the grazing process, like bite characteristics, the bite characteristics within feeding station. Mindy simulate feeding stations, grazing events of meals, and all the way to a scale of paddocks throughout the day or different days. So just to give you an idea of what Mindy can do, here we go to grazing methods, and we have, how the grazing pattern change throughout the day, depending on the grazing method and the type of the cow. The model with the model outputs like go from dry matter intake, rumination time, eating time, but you can ask for particular variables of the model. The model have over 2,000 variables at each grazing event. Again, to illustrate one of the things that the model can do, even if you want to get into the rumen of the cows, and at each particular meal or grazing event, you can go from a behavioral point of view, intake rate, bite rate, even the locomotion dynamics, right? So all the way to what's going on in the rumen and physiologically from or endocrinologically asking for variables like we call the hunger hormone, which represent ghrelin. So this is too detailed already for this early in the morning, at least for me, but this is wanted to show you what the model can do as well. In addition to those sort of things of representing the diurnal grazing pattern at different scales, the model, had the mechanism to represent the post-ingested feedback and also integrated with the minimal total discomfort model of Mike Forbes. So the integration of those two theories into the model enabled Mindy to express desires of what she would like to eat and what she selects based on the constraint that she faced in the grazing scenario. So I'm not going to go through the details in there, but as you have the presentation in front of you, whenever you want, you can go and look at what are the variables that we take into account to determine what Mindy prefers to eat, and based on the constraint that facing the grazing environment, what she selects to eat. But these are typical examples, and I'm not going to go through there, but long story short, what we did with Juan and Fred a while ago when we were developing this model, and just test and evaluate Mindy and see whether Mindy was able to reproduce some of the experiments that Juan and Fred ran a while ago. So even from a diurnal point of view, Mindy can represent or simulate the diurnal changes in preference with a greater preference for legumes in the morning and grasses later on in the day. To continue and just describing this model, in addition to all those rumen grazing behavior variables, if you like, you can ask the model, okay, what is the energy cost of behaving the way she's behaving, from a bite to rumination all the way to walk from one place to the other. So you can not only have a mechanistic account for the maintenance cost, but a mechanistic account for the activity cost or of the grazing cost, if you like. So this is a module that we created to represent the energy expenditure or the rate of energy expenditure by the second. So literally, as the animal, as the Mindy is grazing, selecting any particular plant, you can go from variables that tell you the YATP in the rumen, all the way to how much energy is investing in each particular mastication during the oral processing, if you like. So as you can see, the model is quite detailed and allow us to ask a lot of questions that would be literally impossible for isolating real life. Not going to go through details in there, but Mindy can also, and she does, graze with friends. So it's not a single expression of a cow, but it considers the interaction with different patches in the pasture and different pair size. So these are examples, and I'm not going to go through that, but pretty much in going through this, Mindy can tell you, like I said before, how much energy she's going to invest while she's grazing, whether she grazes alone and she grazes with a couple of friends, or where she's investing more energy at the bite level if she's grazing a tough versus a soft pasture, or where she change the energy expenditure rate at the bite level or step level or minute level or meal level, you name it, if she's grazing a patchy pasture versus an homogeneous pasture. So I'm going through some of the things that it was mentioned before by Jennifer. Here in New Zealand, as you can imagine, because of the color of this antipode island, which is green in the North Island especially, we got a lot of nitrogen and quite a lot that has been translated, for example, here in the Canterbury, in the South Island, with blue babies, with a lot of nitrogen excretion at the urine patch and the urination event and leaching to the waterways. So it was quite important by then to develop the urination and drinking behavior of the model. So we can simulate the urination pattern through MINDI at each urination event and throughout the day, asking as well the volume and the nitrogen concentration of each urination event and daily excretions, if you like. So to give you an idea of the complexity of this model, here we go. So this is the flow chart diagram that start with different plants, if you like, and the chemical composition of those plants attached to the water ingestion and drinking, and all the way to kidney functions and rate of bladder filling, interacting with the blood urea nitrogen. And in that way, we can simulate literally mechanistic urinary nitrogen excretion, in addition to the total of the pattern of that urination behavior. As you can see in this graph, we can simulate, nitrogen load that change throughout the day. These are just different diets, the size, in that particular graph, the volume of each urination event and how it changed throughout the day based on diet. So this is just to illustrate some of the things that can be done with MINDI, or the questions that you can ask me. Last but not least, one of the things that I mentioned, one of the last models that we incorporated to MINDI was the oral processing. So pretty much MINDI process each of the forage that she eat based on the whole interaction of the modules that I mentioned before. So pretty much a bite or cluster of bites in terms of ingested bolus of lucerne is not, alfalfa is not going to be masticated or ingested the same way that she would do if she was consuming maize, for example. So that's the module of MINDI when we talk about in oral processing and rumen digest outflow. And this is quite important, not only from a nitrogen dynamics in the rumen, but also from a fermentation pattern. And in this particular case, I think they are quite interested in the methanogenesis. So literally methane production in the rumen is simulated quite mechanistically through the interactions of all these modules. So implications of using this model is complementary to the reality. It's a tool, it's a heuristic tool that can help us to describe why those things happen rather than only what has happened. We can use this model in this particular case, and then the way we are going to be using is to evaluate different mixers of feeding circuits or time availability, or access to of the animal to those islands and access to particular plants. We can use this model to, I have toilets there, but when the animal is going to eat and what she's going to eat, at the same time of asking MINDI how comfortable she feels when she's off of a particular particular species. In that way, and evaluating, an arrangement, factorial of species, timings, etc., of those species available to the model, you can start thinking of, four-dimensional high definition design of islands and how you present those islands to the animal. So that's the tool that I wanted to present. I must say that the work that is going to be conducted from this model lean point of view, it's mainly going to be run by that postdoctoral fellow of mine. Her name is Conan Garrett. So Juan, I hope I presented what you wanted me to present regarding to the model, but I can develop a little bit more of what we talk about. I'm open to questions now.
Sure, yeah, great. Yeah, there's a question by Rick. Can Mindy model effects of air temperature or climate on animal behavior?
We can, that's a very good question. And Mindy's got weather included there. We can alter some of the variables, especially the variables are related to water dynamics in the model regarding to heat stress and alter, drinking behavior as mechanistically when we vary some of those variables in the model. We cannot, or Mindy cannot simulate the effect of cold stress.
Okay, great. And there's another question by Frida. How is the model 4D exactly? Can you explain a little bit more that 4D type of?
4D, it's a 4-dimension. The model, considers 3-dimensional arrangements, the 3 dimensions of in space and how the forest and biochemicals are distributed in space, and the 4 dimensions time. So the way that the model works, it considers diurnal fluctuations in photosynthesis, transpiration, and evapotranspirations of the plant, which are affected at the seasonal level according to the seasonality of radiation and where the animal is located, latitude-wise, if you like. So all those things affect at the very end of the day in time, the chemical composition of the forest, but in the model, like I said before, although it's not spatially explicit in where the animal is going to be foraged, the actual heritage can be represented in 3 dimensions.
Great, okay. So, yeah, so then we'll keep, I think, your discussion then as we move to this open floor. So we move on the next piece of the puzzle, Seth, which is going to be not only testing these interactions in vitro, in vivo, and in silico, but in vitro. So that's the work by Fernanda Batistel, who is in University of Florida, and she's been looking at interactions between the grass and then these legumes and forbs within in vitro systems. So that's she's traveling today, so she left her presentation recorded. So I think that's so the next section is going to be on how we look at these synergisms and interactions from an in vitro standpoint, ideally looking at the best candidates that we identify in silico. So as we get more constraining capabilities, we start testing, approaching to real life now on an in vitro system.
Hi there, my name is Fernanda Batistel, and today I will be sharing what I will be doing in this project. So basically, I will be responsible for objective 1C for the in vitro test of the combinations of the forages. More specifically here, I will be evaluating how forages and their combinations alter rumen fermentation, microbial growth, and nutrient digestibility. And I will be using an in vitro system called continuous culture fermenters. And I will give you more details about this system in a little bit. If we think about the gastrointestinal tract of ruminant animals, they have a very unique system. And we have 3 extra compartments if you compare with the human gastrointestinal tract. So ruminants, they have the reticulum rumen, omason, and abomason. The abomasum is, although we have another name, is very similar to the stomach, to our stomachs. And the reticulum and the rumen, they are very important because they have a very high concentration of microorganisms, and these microorganisms are the ones that will be doing the rumen fermentation. And these allow them to convert forage or other low quality feeds rich in fiber into high quality proteins such as milk and meat. When you think about the rumen fermentation, once the forage enters the rumen here, we have the microorganisms that will be doing the fermentation, and we have two very important end products when we think about ruminant nutrition. The first one is the production of short chain fatty acids, and the main ones are acetate, propionate, and butyrate. These end products, these short chain fatty acids, they are a very important source of the energy for ruminants. They can give up to 85% of the energy requirement of the animals. And the second one is that these microbes, they will be growing in the rumen, in the reticulum, and they have a short life, or even because of the passage rate, the high passage rate of the rumen, at some point they will be ending up in the small intestine, where we have digestion and absorption of this microbial protein. And the microbial protein is one of the best sources of the protein in the diet because they have a high digestibility, and also they have amino acid profile similar to the amino acid profile of milk protein and also muscle. Another point that I would like to highlight here is the importance of increasing forage digestion, specifically in the rumen. We have a very important impact in animal production. So as we increase the forage digestion, we can increase the feed intake of the animals, we can increase the fat-corrected milk that is very important for dairy cattle because the producers get paid by the amount of fat and protein in the milk. And we also have the environmental impact side here. So as we increase the digestibility, we can decrease the methane production, we can decrease the nitrogen excretion of ruminant animals, and we can decrease the water and land use to produce ruminant animals. So in this picture here, you can see the continuous culture fermenters. They are a system that mimics the rumen of ruminant animals. And if we take a closer look here, you can see that pretty much we control the environment, the inside of this vessel here, this glass vessel, in a way that is very similar to the rumen of a cow. So we have a buffer delivery that mimics the saliva. We have a CO2 port there that will take all the oxygen out because we don't have an oxygen in the rumen. We have a mixing system because in the rumen of the cows, we have a lot of contractions that will be mixing up the feed particles with the microbes. We'll be keeping the temperature at 39 degrees Celsius because it's the temperature of the rumen. And the last thing that I would like to point out here is that in this part here that we usually call outflow is mimics what is leaving the rumen. So the majority of the samples that we'll be collecting will be coming from the outflow. So at the end of this line, we have a bottle, so we collect the samples from there, and then we can evaluate, for example, the nutrients to see the digestibility inside of the rumen. Our experimental design is 8 by 8 Latin square design. This means that we have 8 fermenters, and all the diets, we will have 8 fermenters and 8 diets, and all the diets will be going over throughout the time to all the fermenters. The periods will be 6 days of adaptation and 4 days of sampling. This indicates that at the beginning of each period, we'll be going to the dairy farm. We'll be collecting rumen fluid from 2 cows, and we'll be incubating in all the fermenters. We'll start to feed the specific diets, and after 6 days of adaptation, then we'll be keeping the same things. We'll be doing the same thing as we did before in the 6 previous days, but in this part here, we'll be also collecting samples. That is why we call sampling period. So we'll have 8 replicates by each treatment, and a total of 64 observations. So for our treatments, we have a control diet or a basal diet that will be a grass. And then for the other treatments, we'll be using the basal diet, 85% of the basal diet. In the 15%, the other 15% will be having a combination of the legumes and forms, and we'll be seeing their effects on the on-rumen fermentation. We'll be analyzing the samples for short-chain fatty acids that, as I mentioned before, they are a very important source of energy for the animals. We'll be analyzing the ammonium nitrogen. That is important because it will give us an idea about the nitrogen metabolism in the rumen. We'll be analyzing the microbial growth. That is very important, a very important source of microbial protein for the animals. We'll be analyzing the nutrient digestibility, so how much of the diet will be digested inside of the system, inside of the rumen. And we'll also be analyzing specific nutrients such as fiber, protein, starch, and fatty acids. And for the last 2 things here, we'll be analyzing the total gas production that will give us an indirect measurement of the fermentation. So if we increase the gas production, that indicates that we have a higher fermentation. And the methane production here also will be important to see if one or more diets decrease the methane production. That is also something measurement very important. So ideally here, we hope to see that some of the combinations, the forages combinations, will increase the short chain fatty acid production, will increase the microbial growth, will increase nutrient digestibility, and will decrease the methane production. So that is a good thing to expect from these combinations, because previous studies, they observed that when we have legumes and grasses together, we tend to enhance the animal performance. So we hope to see something similar here. The data will be analyzed using a mixed model. So we'll be using the random effect of a period and fermented, and the fixed effect of treatment. So I will do my best to make our meeting in person tomorrow. But if I cannot make and you have any questions or suggestions, please send me an email, and I'll be very happy to work with you. So I hope you have a great day, and thank you very much.
Great. So yeah, Fernanda is not here, as I mentioned. She's in Brazil now, but so if you have any questions, or I don't see anything in the chat, so let's move to, yeah, keep filling our puzzle. So Seth, if we bring that slide on our next presenter that, once we identify those combinations that Fernanda and Pablo were mentioning, that's the next objective, is to deploy those islands in the landscape. So the idea is how we spatially distribute those islands in the landscape, and then and how we visualize those. So we have, the first is the spatial arrangement, how you put those islands in the landscape, and that's Doug Ramsey is going to present from Utah State. A great idea he had on that approach for deploying those islands in the landscape as opposed to doing it randomly. So Doug, let's upload your your slide and that your
thank
idea.
you. And still have to find a thing. Pardon me, I'm glad to be here. So let me make some conversation before I can find this thing. There we go. Glad to be here with all you old friends and mentors and new friends. And it says I can't share. Let's try that again. Okay. I'm just going to keep it like this because I know that that Zoom might mess up with my different screens. What Juan, when he approached me last year, wanted me to identify potential sites or locations for these islands. And when he described the project to me, it was essentially within a given landscape, an enclosed landscape, a pasture, where could we put these foodscapes? The ideal thing, of course, is just distribute them evenly, but landscapes aren't even. So we had to find a way to identify where the best place was so that these islands, the vegetation that goes in these islands would have the best chance to grow. So the idea that I came up with was to take imagery, satellite imagery mostly, though I think at the scale we're talking about, we might have to move down to drone imagery. But in this case, I'll just show the satellite imagery. But to take this imagery across a number of years and identify within the pasture those areas that exhibit consistent above average response with vegetation. So essentially find the places on a yearly basis that are greener than all the other places on the pasture, assuming that those greener places have more resources to become green. So more moisture around here mostly. So this is a function of identifying the areas that have the most moisture availability, which is exemplified by the amount of greenness on the ground on a yearly basis. So to do this, I'm using Google Earth Engine, and I've been using Google Earth Engine for a couple of years now and developed a number of tools to monitor landscape. If you're not familiar with Google Earth Engine, it's currently the cool image processing tool du jour, right? So tomorrow there might be something different, but right now it's a thing that a lot of people are using to monitor landscapes. And this platform, the whole Google verse, if we call it that, if you can call it that, stores a number of petabytes of imagery and other data layers that on a global basis that we can use they're almost at our fingertips. So it reduces the amount of data collection and processing we have to do by a few million times. So we can look at multiple sets of imagery within minutes as opposed to within weeks that we did in the past. So it's a very nice platform. Other than the scale that we're working with, it works really well with what we're doing here, and that's to identify areas that have some positive aspects to allow vegetation to grow a little bit better than other places. So the basic approach I was taking is to look at imagery for the same pasture. In this case, this is a sample pasture located here just north of Logan and Richmond, where I've collected imagery through Google Earth Engine for the past 7 years. And within those 7 years, just selected 1 month, in this case, August. And within August, and for those years, calculate the average greenness of that pasture. And then for each year, identify the pixels that are above average. So they have more vegetation. So why August? I have there. When I've tried to monitor the landscapes and select a specific part of the year to monitor with, I found that late summer imagery tends to give a better idea of what's happening on the ground than, say, spring imagery, where things are really green, mostly because there's such a stochastic nature to spring. We don't know if the response from the sensor is a function of what's happening on the ground or whether it's just some weather anomaly that happens. So we had a lot of rainfall one spring and it gets way too green above normal. So in order to map or to monitor month to month to month, I tend to gravitate towards late summer. I could easily choose July, but if I go earlier than that, I might get that oscillation that I really don't want, because I don't want anything outside what's on the ground to affect what's going on. So August, everything tends to settle down a little bit. So I calculate the average for a certain set of images. In this case, just August for 2021. There are 6 images there, and I can take those 6 images and generate one image that represents all of the entire month of August. This is mostly done to reduce clouds and bad pixels, things like that, just to get a clean image of the surface for a certain period of time. Oops. Once that's done, and this is relatively short presentation because I don't have a field site to work with yet, so this is all conceptual and relatively simplistic. Depending on the site, we might need to, if it's available, have soils data, topographic data at the appropriate scale and so forth. But in this case, just looking at greenness and looking at how that greenness changes across space and time, we can take that pasture, which you see on the left-hand side here, and take the different years. So in this case, 2016 through 2021, so 5 years of data. And each one of these represents the amount of greenness on the ground for that year, of which I calculate the average greenness for this entire area and the standard deviation. And using those, I identify those pixels within that particular landscape that are on average higher than other pixels. The things that are lower on average don't get collected. So my break was 1 was the mean plus 1 standard deviation. So just the one side, the greener part. Everything less green than that, I eliminate. Taking those images, I can just calculate the frequency of times each pixel is above average. And that represents the image in the middle, which shows you the number of years coded from dark blue to red. So dark blue would be only one of those 5 years it was above average, and red represents all 7 years were above average. And you notice that the reds kind of hover around the edges, particularly in that little orchard patch there, mostly because there's irrigation going on there. This is a non-irrigated pasture, except for those small parts. But this provides an idea of which sites are greener, more greener on average on a yearly basis than other areas. And it's mostly a function of topography, because if you look at the this primary area here, which is full of above average pixels, it basically follows a drainage. So these are just the low spots on the ground. So you can use it as a indicator of concavity, how concave the landscape is, and therefore its ability to catch water and store it. So after we do this, the other problem is where to locate it spatially. So these green patches here might be potential sites to locate islands of these legumes. We haven't gotten to that part yet to identify exactly where and what the distribution should be, because we don't know exactly where we're going to be dealing with yet. So I'll leave it at that. We're also going to be working, I'll also help to work with the location data. So the collared animals, their GPS data to identify what their spatial distribution is and how they move through the landscape and interact with these islands. Thank you.
Great. Yeah, that's any questions? I have one, why 7 years is, so you think that's the ideal time frame, or can be more or less?
We can do more. I use 7 years because this particular data set, which is a Sentinel-2, the European Space Agency satellite, is only available for 7 years. I could use Landsat, which we can do the last 30 to 40 years. The resolution is a little bit lower there, but that's about it. So we can do any timeline we want. I thought that using too large of a time period might introduce too many trends in the landscape and other management activities. Doing a shorter period of time kind of keeps the management anomalies at bay a little bit.
Great. Yeah, and then yeah, something discussing later on, but that's a question from Rita. Would you not also put some foodscapes in the dry areas or intermediately dry areas to compare?
That would depend on how well the plants can survive in dry areas. So the idea here was to identify areas where plants could be easily established. If the plants can survive in dry areas, and that's what we need to figure out. If we have an idea of the plants we're going to be using and their ability to survive in dry environments, sure, we can do that.
Yeah, and that should be also linked with what Jennifer presented early on in terms of gathering information from those plots on establishment and persistence. So yeah, okay. So Tim is asking, in some instances, is Sentinel-2 is too coarse for some pastures? Is a plan to fly a drone for multiple flights in the month of August? How does that work going forward instead of using historic imagery monitoring?
We will have to use drones in these pastures just because they're too small, and from that develop some surface models to identify areas of the depression areas that could be more moist than other areas, or provide a better habitat for the plants. The only reason I use Sentinel is because I was looking backwards, trying to identify locations in general. So Sentinel in this particular pasture worked out just fine as a coarse tool to just triage locations that we'd have to look at more closely. But yes, we'll have to use drones in the future.
Great. Okay. So yeah, and then we'll leave all these other questions for our open floor. So we are really on time here. So the plan was for the coffee break at 10:40 and we're at 10:37, so that's great. So yeah, let's take much needed coffee break now until 10:55, where we resume our sequence of presentations. We have a little bit more time. That's we're really on time for this, at least for this part of the presentations.